Welcome. Today, we're diving into the world of endotoxin testing and pyrogen analysis. I'm Dr. S.K. Singh, and together we'll explore the key concepts, methods, and importance of detecting these harmful substances. Let's get started and uncover why this area matters so much in healthcare and safety. Let's start by understanding what pyrogens are: substances that cause fever when they enter the body. They matter a lot because they impact patient safety, product quality, and regulatory compliance. Sterility assurance depends on controlling these pyrogens. There are two main types: endotoxin pyrogens, which come from gram-negative bacteria and are mostly lipopolysaccharides, and non-endotoxin pyrogens. These include components from gram-positive bacteria, fungi, viruses, and even chemical contaminants. Knowing the difference is key to managing risks effectively. Let's talk about endotoxins, specifically their structure and where they come from. Endotoxin is part of the outer membrane of gram-negative bacteria, and it's made up of three main components: the O antigen, the core polysaccharide, and lipid A, which is the part that actually triggers fever. You'll find endotoxins lurking in water systems, raw materials, manufacturing equipment, and just about anywhere there's environmental contamination. They're tough to get rid of. Normal sterilization won't cut it. To break them down, you need really high heat, around 250 degrees Celsius for at least half an hour. Let's take a look at global regulatory expectations around endotoxin testing. Different regions and organizations have their own guidelines, but they all aim for the same goal: ensuring product safety. The United States Pharmacopoeia sets standards like USP 85 for bacterial endotoxins and USP 151 for pyrogen testing. Europe's Pharmacopoeia and Japan's guidelines offer similar tests. Meanwhile, the FDA provides guidance on validating these methods, and international bodies like ICH and the EU's GMP Annex 1 set specifications and controls to keep endotoxins in check. Endotoxins pose serious clinical risks, starting with symptoms like fever, chills, and low blood pressure. In severe cases, they can lead to septic shock and even organ failure. Certain products carry higher risks: intravenous injections, large volume parenterals, dialysis solutions, biologicals, and vaccines. For example, if an IV solution is contaminated with endotoxins, it can cause a sudden and intense fever reaction within just a few hours. Understanding these risks helps us prioritize safety in critical treatments. When it comes to detecting pyrogens, there are several key methods we rely on. First, the bacterial endotoxin test, or BET, uses a substance called limulus ambosite lysate, derived from horseshoe crab blood. This test comes in three forms: gel clot, turbidimetric, and chromogenic, each detecting endotoxins in different ways. Then there's the rabbit pyrogen test, which measures fever response directly in rabbits. Finally, the monocyte activation test can detect both endotoxin and non-endotoxin pyrogens. Importantly, regulators are pushing for alternatives to animal testing, shaping the future of pyrogen detection. Let's look at the advantages of this method. It's simple and cost-effective, making it accessible for many labs. The test uses a compendial approach, meaning it's standardized and reliable. Here, how it works: endotoxins react with the LAL reagent, and if a gel clot forms, that's a positive result. No clot means negative. The procedure is straightforward: mix your sample with the reagent, incubate it at 37 degrees Celsius, then check for clot formation. It's a clear, easy-to-interpret test. Let's look at two key methods for detecting endotoxins: the turbidimetric and chromogenic approaches. The turbidimetric method works by measuring how clot formation, triggered by endotoxins, makes a solution cloudy. We track this cloudiness by changes in absorbance. It's commonly used for routine quality control in injectables and water systems. On the other hand, the chromogenic method relies on an enzyme cascade activated by endotoxins, producing a yellow color we measure with a spectrophotometer. This method is quantitative, automated, and highly sensitive. Both have their place in ensuring safety. Now, let's talk about how we calculate endotoxin limits. This step is crucial because it sets the maximum amount of endotoxin allowed in a product to ensure safety. We base this limit on factors like the product's intended use, the dose, and the route of administration. Getting this calculation right helps us protect patients from harmful reactions while maintaining product quality. It's a careful balance and a key part of our overall safety strategy. Now, let's talk about maximum valid dilution, or MVD. This calculation helps us understand the highest level we can dilute a substance while still reliably detecting it. It's a crucial step for ensuring accuracy in measurements, especially when working with very low concentrations. By knowing the MVD, we avoid wasting resources on dilutions that won't give us meaningful results. It's all about finding that sweet spot, the balance between sensitivity and practicality. Now, let's talk about method validation. This is where we make sure our approach actually works, that it's reliable, accurate, and fit for purpose. Validation confirms the method's performance meets the standards we need. It's a critical step before we can trust any results or make decisions based on the data. Without it, we're just guessing. So, method validation is the foundation that gives us confidence moving forward. Now, let's focus on endotoxin testing for pharmaceutical water, a critical step to ensure safety and compliance. This testing applies mainly to purified water and water for injection systems. For water for injection, the endotoxin limit is very strict, no more than 0.25 endotoxin units per milliliter. Dialysis water limits vary, depending on the pharmacopoeia standards. Here's an example. A measured result of 0.12 EU per milliliter falls well below the 0.25 limit, so that's a clear pass. When it comes to Pyrogen investigation and handling out-of-specification results, we start by looking at potential sources. These include raw materials, water systems, equipment contamination, cleaning failures, and environmental factors. Our investigation follows clear steps. First, we confirm the result itself, then review the analyst's technique and the instrument's qualification. We also examine manufacturing records and evaluate where contamination might have come from. Finally, corrective actions might involve optimizing depyrogenation cycles, sanitizing water systems, or improving cleaning validation. It's a thorough process to keep things safe and compliant. When regulators come knocking, they zero in on a few key areas. They'll review documentation closely, method validation reports, endotoxin trend reports, investigations into out-of-spec results, and water system monitoring records. Common shortcomings pop up here, like inadequate inhibition studies, insufficient validation, weak trend analysis, and poor contamination control. Annex 1 raises the bar, emphasizing that endotoxin control must be fully integrated into the overall contamination control strategy. This integration is critical for meeting today's expectations. Let's wrap up with a quick summary and the key references behind today's topic. Endotoxins are heat-stable pyrogens, mostly from gram-negative bacteria, and their contamination can cause serious reactions in patients. Testing standards like USB, EP, and others set clear requirements. The LAL test remains the industry standard, but the MAT offers broader detection. Reliable results depend on proper validation and careful study of interference. In the end, strong endotoxin control programs are vital for keeping patients safe and meeting regulations worldwide. Thanks for joining me today. If you want to dive deeper or have any questions, don't hesitate to reach out. I'm here to help. Until next time, take care and thank you for your attention.